Hello friends, welcome to 10 out of 10 Smart Biology. Today in this video lecture we will be discussing post fertilization events. हमने previous video में process of fertilization को in detail describe किया. In this lecture we will discuss what happens to the zygote after fertilization. Okay, all the events that take place after the process of fertilization are encompassed under the heading post-fertilization events. Okay, so let us see what happens when fertilization takes place. जब fertilization हो गया, after some 24 to 30 hours after fertilization होने के 24 to 30 hours after fertilization, जो zygote है, okay, zygote अभी जो है वो कहाँ fallopian tube में है. एम्प्यूला इस्थमस जंक्शन जो है वहां पर फर्टिलाइजेशन हो गया. अब ये ज़ायगोट स्लोली मूव कर रहा है. इस मूवमेंट के दौरान आफ्टर 24 आवर्स ऑफ फर्टिलाइजेशन ज़ायगोट जो है इट अंडरगोज डिवीजन, इट अंडरगोज अ स्पेशल काइंड ऑफ माइटोसिस. ओके, एंड दैट स्पेशल काइंड ऑफ माइटोसिस इज कॉल्ड एज cleavage. Fine, that's called as cleavage. Now, what is cleavage and what is the difference between mitosis and cleavage? इसको cleavage क्यों बोलते हैं और mitosis और इसमें क्या difference है? देखिए, जब mitosis होता है, when mitosis occurs, okay, before entering mitosis, a cell, it undergoes certain phases. Number one, called as G1 phase or growth 1 phase. पहले सेल जो है G1 phase में होता है and in G1 phase a cell decides whether it has to divide or not. When it decides it has to divide, okay, it grows in size and then it shifts, it enters another cycle, another phase called as S phase. S phase में chromosomes duplicate हो जाते हैं. Then the cell enters the G2 phase जहाँ पर growth हो जाता है, सेल का size increase होता है, mRNA content increase होता है, proteins increase होते हैं. Then the cell undergoes mitosis. लेकिन जो cleavage है, it is such a type of division, mitotic division, where there is no G1 phase, where there is no G2 phase. So ये ऐसा mitosis है जिसमें growth phases और gap phases absent होते हैं, सिर्फ S phase होता है. That's why जब क्लीवेज होता है, जो सेल डिवाइड होती है, इसका साइज जो है वो सिमिलर रहता है एंड द नंबर ऑफ सेल्स इंक्रीजेस. सेल्स का नंबर इंक्रीज होता है लेकिन सेल्स का साइज स्लोली डिक्रीज होता जा रहा है. ओवरऑल जो पैरेंट सेल का साइज था वो कांस्टेंट रहेगा लेकिन जो नई सेल्स बनेंगी उनका जो साइज स्मॉलर होगा. सो हम कहते हैं क्लीवेज क्या है? क्लीवेज इज अ रैपिड माइटोटिक डिवीजन विदाउट ग्रोथ और गैप फेजेस. सो व्हाट इज क्लीवेज? इट इज रैपिड माइटोटिक डिवीजन. व्हेन अ सेल undergoes rapid mitotic divisions without growth phases, such a kind of division is called as cleavage. And we must remember that cleavage occurs only in human beings. It occurs only during embryonic development. It occurs only when the zygote divides to form a particular structure. Okay, and now in cleavage what happens? What is the result of cleavage? In cleavage there is increase in cell number without increase in cell size. So cells जो बन रही हैं, डॉटर सेल्स जो बन रही हैं, उनका साइज डिक्रीज होता जा रहा है लेकिन उनका नंबर इंक्रीज हो रहा है. जो पेरेंट सेल जायगोट थी उसका जो कांस्टेंट अपना साइज वो वही रहेगा लेकिन जो वहां से सेल्स बनेंगी उनका सेल साइज जो है डिक्रीज होगा. राइट? So when does first cleavage occur? पहला क्लीवेज कब होता है? फर्स्ट क्लीवेज जो है जब zygote बन गया, फर्टिलाइजेशन हो गई, zygote बन गया. When will first cleavage take place? First cleavage takes place 24 hours after fertilization. Okay? Fertilization जब होगी उसके बाद 24 घंटों के बाद. Some say after 24 to 30 hours, but the average time is 24 hours. After the 24 hours of fertilization, the zygote undergoes first division, first cleavage. और ये जो division होता है, when in human beings the zygote divides after 24 hours of fertilization, this division is not equal, it's unequal. जो सेल्स जो zygote डिवाइड होता है, दो सेल्स में डिवाइड होता है लेकिन ये दो सेल्स जो है इक्वल साइज की नहीं बनती. एक थोड़ी सी बड़ी बनती है, एक थोड़ी सी छोटी बनती है. यहां पर कुछ टर्म्स है which we must know. In human beings the cleavage is number one, it is holoblastic. It is holoblastic. Holoblastic means that there is complete division of cell. There is complete division of cell. There is complete division of cytoplasm. See, basically there are different types of eggs on the basis of amount of yolk. There are certain eggs in which there is a lot of yolk. Such eggs are called as polylecither or megalecither. For example, the egg of hen. अब hen का जो egg है इसमें बहुत सारा yolk है, तो most of the space of the egg is occupied by the yolk और बहुत कम रीजन ऐसा होता है जहां पर yolk नहीं होता है. तो when such a type of egg, polycystal egg divides, yolk cannot divide. Only that portion of the egg divides which is free of yolk, and that is a little portion. Such a type of cleavage will be called as Neuroblastic Cleavage. But in human beings, we know that there is too much of negligible amount of yolk. Okay, negligible amount of yolk, as if nothing, equal to nothing, जैसा कुछ है ही नहीं, इतना कम योग है, माइक्रोलिस्टल एग है. तो वहां पर जो है जब डिवीजन होता है तो अक्रॉस द साइटोप्लाज्म होता है, पूरे सेल के आर पार डिवीजन होता है. व्हेन द सेल डिवीजन इज कंप्लीट, okay, the complete cytoplasmic division occurs, such a type of division is called holoblastic, okay, complete division. But we must remember that in human beings, okay, the division cleavage, first cleavage is holoblastic, but it at the same time it is unequal. That is, when the cell divides, it does not divide into two equal cells. It's unequal. When the cell divides, a cell is one of the daughter cells is a bit smaller and one of the daughter cell is a bit larger. There is one more important part. In human beings, the cleavage is indeterminate. In human beings, the cleavage is indeterminate. That is, when a cell divides, okay, when the zygote divides into two daughter cells, the fate of these cells is not destined. The fate of these cells is not fixed. मतलब हुआ जब zygote ने डिवाइड किया, दो सेल्स बनी, अब ये वाला सेल क्या बनाएगा, एम्ब्रियो का क्या पार्ट बनाएगा, ये वाला सेल एम्ब्रियो का क्या पार्ट बनाएगा, ये अभी फिक्स नहीं हुआ है. When the fate of the cells formed due to cleavage is not fixed early, such a type of cleavage is called as indeterminate. Okay, and this indeterminate cleavage is characteristic feature of what? It is characteristic feature of deuterostomes. It is characteristic feature of deuterostomes. You know deuterostomes? Deuterostomes are those animals in which anus develops first during embryonic development. Well, there is another group of animals called protostomes. Protostomes are those organisms, those animals in which mouth develops first during embryonic development. Okay, so cleavage is indeterminate in case of humans, and deuterostomes including echinodermates. Echinodermates में भी क्लीवेज जो है indeterminate है. लेकिन जो प्रोटोस्टोम्स होते हैं उनमें जो क्लीवेज होता है that is determinate. That is, as soon as the zygote divides and the number of cells are formed, say when zygote divides, it forms two cells. Those two cells will again divide with four cells, and the fate, the future of those four cells is fixed early. वहां पर फॉर एग्जाम्पल अर्थवर्म में zygote ने 4 सेल्स बनाई, तो अर्ली एम्ब्रियोनिक फॉर्मेशन में ये 4 सेल्स में सेल नंबर ए जो है पहले से फिक्स होगा, सेल नंबर ए हेड बनाएगी, सेल नंबर बी जो है कोई दूसरा पार्ट बनाएगी, सेल नंबर 3 नेफ्राइड्स बनाएगी, सेल नंबर 4 कुछ और बनाएगी. तो फैट जो है अर्ली जो फिक्स होता है. अगर इस कंडीशन में क्या हुआ, सेल ए डैमेज होगी तो अर्थवर्म बनेगा लेकिन विदाउट हेड बनेगा, फॉर एग्जाम्पल. सो such a type of division is called determinate. So there are two types of cleavage, indeterminate and determinate. Indeterminate cleavage is that cleavage in which the fate of the cells is not fixed early, whereas determinate cleavage is that cleavage in which the fate of the cells is determined fixed early, right? Okay, so पहला क्लीवेज जब होता है it occurs in the animal vegetal axis. वो हम पहले ही डिस्क्राइब कर चुके हैं जब हमने फर्टिलाइजेशन की बात की कि एग के दो एग इज अ पोलर स्ट्रक्चर, एनिमल पोल होता है, वेजिटल पोल होता है, स्पर्म जो है एनिमल पोल से आता है, स्पर्म की एंट्री थ्रू एनिमल पोल हो जाती है, एंड दैट पाथ ऑफ एंट्री, ओके, इट इज बेसिकली द फ्यूचर प्लेन ऑफ डिवीजन. इसी प्लेन में एग जो है zygote जो बाद में डिवाइड करता है. तो पहला क्लीवेज जो होता है तो किस डायरेक्शन में होता है, किस प्लेन में होता है, इट ऑकर्स इन द एनिमल एंड वेजिटल पोल एक्सिस, एनिमल एंड वेजिटल पोल axis. So this is how the process of cleavage takes place in human beings. Okay, after the cleavage there is one more event, post fertilization event, and that event is called as formation of Marula. Formation of Marula. Second जो cleavage जब होता है it results in the formation of a structure called as Marula. Marula क्या है? Marula is a solid ball of cells which looks like mulberry. जैसे mulberry का fruit होता है, बिल्कुल जब zygote under division हो जाता है, when it undergoes division, rapid mitotic divisions, it leads to the formation of a ball of cells, a solid ball of cells, okay, and that solid ball of cells resembles mulberry. That's why it's called Marula. Marula is a Latin term derived from morus. Morus means mulberry. Okay, now let's see how does this occur. Fine. See, जो ये हमारे पास जायगोट है, जायगोट के बाहर विट्रोलाइन मेम्ब्रेन है, जायगोट के बाहर जोना पेलुसिडा है, जायगोट के बाहर कोरोना रेडिएटा है, सारे लेयर्स मौजूद है. ओके, ये जायगोट जो है आफ्टर 24 आवर्स of fertilization, जब fertilization के 24 घंटे गुजर जाते हैं, then what happens? This zygote undergoes first cleavage division. It undergoes first mitotic division, and the division is holoblast but unequal. तो दो cells बनती हैं. Here we get two cells. One of the cells is smaller in size, and one of the cells is larger in size. These two cells, which are formed as a result of first mitotic division or first cleavage division, are called as blastomers. Okay, so what are blastomers? Blastomers are the cells which are formed as a result of cleavage of zygote. Fine. Okay, so now after this again we must remember that zona pellucida, vitelline membrane, corona radiata सारे के सारे layers intact हैं अभी. Fine. इसके बाद what happens? Since the larger, इसके बाद दूसरा division होगा. After that's again after some 24 to 30 hours. अब पहला cleavage जो है, first cleavage division occurred after 24 hours of fertilization. Second cleavage division also occurs after 24 hours or 30 hours of the first division. Fine. Okay, तो होता क्या है, जो बड़ी सेल है ना, it divides first. The larger cell divides first and the smaller cell divides a bit later. So in between we come to see a stage called as three-celled stage. Three-cell stage. What is three-cell stage? Three-cell stage is a stage in which arises because of early division of the larger blastomere. Okay, then immediately अब three-cell stage बहुत ज़्यादा नहीं है, it's a transient stage, बहुत कम टाइम के लिए रहता है. The smaller blastomere also divides. छोटा वाला blastomere भी divide होता है and it results in the formation of 4 cells. टेट्रॉड, 4 सेल्स वाला हमारे पास स्ट्रक्चर बन जाता है. ओके, देन this 4 cell structure again goes third division. It undergoes third cleavage division and it leads to the formation of 8 cell. Okay, then 8 cell stage वाला जो हमारे पास स्ट्रक्चर है, that again undergoes rapid cleavage. Okay, and it leads to the formation of 16 cells, a structure of 16 cells. तो 16 cell, this 16 celled structure which resembles, this 16 celled structure resembles a mulberry, and this 16 celled structure is called as morula. This is called as morula. Get my point? So what is morula? Basically NCERT writes it is 8 to 16 cell stage. We must remember, some books write that 16 to 32 cell stage is called as morula, but we must stick to what NCERT says. NCERT says 8 to 16 celled structure is called as morula. Particularly 16 celled structure is called as morula. So 16 celled structure is a ball of cells, a solid ball of cells. There is no space. Okay, there is no cavity. This structure resembles the mulberry fruit, and that's why this is called as morula. And morula has all these layers intact. It has zona pellucida, it has vitelline membrane, zona pellucida, and corona radiata. Now, this 16 cell state, it moves towards the uterus. It moves towards the uterus, and it is said on the third day. On the third day, this morula reaches to the uterus. थर्ड डे में मोरुला जो है कहां पहुंच जाता है? इट इज रोलिंग एंड ऑन द थर्ड डे आफ्टर फर्टिलाइजेशन के 3 दिन के बाद मोरुला जो है इट रीचेस द यूट्रस. व्हेन द मोरुला रीचेस द यूट्रस, इट अंडरगोज अ प्रोसेस कॉल्ड एज कंपैक्शन. Compaction क्या है? अब हम जानते हैं, in morula there are 2 types of cells. Certain cells are smaller in size and certain cells are larger in size. The cells which are smaller in size are called micromers, and the cells which are larger in size called as macromers. Now the morula जो है it undergoes a process called compaction. What is compaction? जो smaller cells है, the smaller cells form the periphery of this structure. Smaller cells, छोटी cells जो है कहां चली जाती हैं? Towards the periphery. And the larger cells are macromeres. They occupy the center of this structure. This process in which these micromers and macromers get rearranged. Micromers form the periphery of the morula and macromers form the center of the morula. This is called as compaction. And during this process, what happens when the morula is undergoing compaction and it is reaching the uterus? During this process, what happens? The cells of corona radiata are lost. Cells of corona radiata are lost, but vitelline membrane and zona pellucida, they are intact. So this is how the morula is formed. After the formation of the morula, when the morula reaches the zygote, Okay, some different events take place. Those events are grouped under the head formation of blastula or blastocyst. तो second stage जो है post fertilization events में जो second या third stage है वो क्या है? Formation of blastula or formation of blastocyst. Now see, on the third day, when the morula reaches the uterus, what does the morula do in the uterus? होता क्या है? मैंने कहा number one, compaction होता है, cells arrange होती है, macromores towards the periphery and macromores towards the center. इसके अलावा हम जानते हैं during this period uterus is prepared. The endometrium is highly glandular and thick, 5 millimeters thick, highly glandular, vascular. And there is one more point that endometrium में कुछ cells होते हैं जो fibroblasts होती हैं. They become glycogen laden. Glycogen accumulate हो जाता है and they secrete various secretions. And those secretions constitute a particular secretion called as uterine milk. Uterine. So endometrium की जो cells हैं, endometrium की जो glands हैं, the glands and the cells of endometrium, they secrete a viscous nutritive fluid. That viscous nutritive fluid is called as uterine milk. Now, अब जो morula on third day, when the morula reaches the uterus, the cells of the morula, okay, they absorb this uterine milk. The cells of the morula, they absorb this uterine milk. जब uterine milk जो है morula के अंदर चला जाता है, what happens? This milk, this uterine milk, this fluid, it builds a pressure. It builds a pressure, and when it builds pressure, ये क्या काम करता है? जो macromores थे, it presses them against one pole. It presses them against one side. बात समझ आ रही है ना? ये compaction हुआ है, morula है, solid ball of cells है. जब ये uterine milk को secrete absorb करता है, जब ये यूट्राइन मिल्क को एब्जॉर्ब करता है और खुद भी ये सेल अपना कुछ सिक्रीशन जो सिक्रीट करती है, तो मरोला के अंदर क्या होगा? फ्लूइड जमा होगा. ये फ्लूइड जब जमा हो जाता है, when the fluid accumulates, it presses the macromores towards one pole. ये जो macromores अंदर थे, इनको ये फ्लूइड एक जो है पोल की जानिब जो है प्रेस करता है, पुश करता है. And as a result, what happens? A large cavity appears inside the morula. अब morula के अंदर एक cavity प्रोड्यूस हो जाता है. Now इसको morula नहीं कहेंगे, because it no more looks like a mulberry. Now its size has increased. Its size has till morula इसका size बिल्कुल उतना ही था जितना zygote का size था. Now because of absorption of uterine milk, the size of this morula changes. Now it becomes a fluid filled ball. Okay, and there is a cavity भी अंदर जो पैदा हो जाता. Now it becomes a ball with a cavity inside. That cavity is called as blastocele. ओके, एंड ब्लास्टोसील में क्या है? फ्लूइड है. यूट्राइन मिल्क ऐसा, ब्लास्टोसील के अंदर फ्लूइड है. फाइन. नाउ दिस फ्लूइड फिल्ड स्ट्रक्चर जिसमें दो लेयर्स है, एक बाहर वाला लेयर ऑफ सेल्स है, एक अंदर जो है मास ऑफ सेल्स टुवर्ड्स वन पोल है. दिस स्ट्रक्चर इज कॉल्ड एज ब्लास्टोसील. This is called as blastocele. So morula absorbs uterine milk and a cavity appears. That cavity is called blastocele, and this structure is called as blastocyst or blastula. This is called as blastocyst or blastula. Now, one more thing, जो peripheral cells थी, they become more flatter. The outer cells they become more flat. Okay, they become more flat, and they are called as trophoectoderm. ये जो outer cells है, this layer of outer cells, ये flat बन जाते हैं ये cells, and they constitute a layer called as trophoectoderm or trophoderm or trophoblast. Okay, now fine. जो अंदर सेल्स है, मैंने कहा वो एक पोल पे चली जाती है. दिस mass of cells which gets arranged on one pole of this blastula, these cells are called as embryo proper. Now ये वाला एरिया, ये वाली cells जो है, they constitute embryo proper, or they are also called as inner cell mass, or they are also called as embryonal knob. This side is called embryonal knob, or these cells form the embryonal knob. The opposite side of the blastula is called as embryonal side, or is called as embryonal end. Fine. So there are two ends in the blastula or blastocyst. One is embryonal end or embryonal knob on which the inner cell mass lies, and the side opposite to this is called as embryonal side or embryonal end. Fine. That's one more important point regarding blastula or blastocyst, that is the cells of the trophoectoderm which are in contact with the embryonal knob or inner cell mass are called as cells of Robber. और कॉल्ड है सेल्स ऑफ रॉबर। और यहां पर हमको और एक चीज याद रखनी है कि जब ये ब्लास्टुला ब्लास्टोसिस्ट यूट्रस के साथ इंप्लांट होगा, यही सेल्स ऑफ रॉबर जो है इसी साइड में, या एम्ब्रियोनल नॉब जो है इसी साइड से, सो द ब्लास्टोसिस्ट गेट्स इंप्लांटेड, इट गेट्स अटैच्ड विद द यूट्राइन वॉल through the cells of rubber or on the side of embryonal knob or inner cell mass. Fine. So this is about the formation of blastula and blastocyst. In the next video, we will be talking about the process of implantation. Thank you very much.